Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen. Scott is taking a break today. Last week we talked to Dr. Nick Matzke, a scientist who has spent decades working on the bacterial flagellum. In addition to giving us a quick introduction to that protein, basically a Flagellum 101 lecture, he also helped demystify some things about proteins, how they can do amazing things like self-assemble, and how they actually work like machines. And he also set the stage for a very provocative paper that he and his colleagues wrote about a possible origin story for the bacterial flagellum. Today we'll talk to those colleagues about that provocative story. I already introduced Nick last week, including how he's bringing together genetics, mathematics, statistics, and various forms of logic, and even geography to trace through evolutionary history. Also here with us in the studio is one of his co-authors on that paper, Dr. Mark Palin, who obtained his PhD at Imperial College and an MD degree at the London Hospital Medical College, where he focused on bacteria and the enzymes that they use to mediate various human diseases. And now he's group leader at the Quadram Institute and professor of microbial genomics at the University of East Anglia in the United Kingdom. And he also is turning his expertise in genetics, microbial pathogenomics, and bioinformatics to studying bacteria and bacterial disease. And then a third guest today is Dr. Matthew Baker, who obtained his D.Phil in physics at Oxford University as a John Monash Scholar, Australia's most prestigious national postgraduate scholarship award. And during that time, he focused exclusively on the bacterial flagellum. And then he was recruited to the School of Biotechnology and Biomolecular Sciences at the University of New South Wales in Australia, where he continued his studies of individual flagellar motors from E. coli bugs using biophysics and fluorescence imaging. So that's just a short introduction to our three experts from three different labs around the globe. Mark in the UK, Nick in Australia, and Matt in New Zealand, doing completely different kinds of research. Mark working mostly in genetics, Nick using computers to sift through massive genetic and protein databases, and Matt actually picking out individual flagellum motors and gluing probes onto them to see how they work. And we got these three together to discuss their understanding of the evolutionary origin of the bacterial flagellum. And if you ask me, they tell a very compelling story. The problem is, you really need a doctoral degree to keep up with them here. I tried to keep the conversation at a non-expert level. I summarized what they were saying, trying to bring it down a few notches with the technical jargon, and tried to avoid the scientific weeds, and I dropped in a number of inserts to explain certain concepts and definitions. And then I asked two people to listen to the edited version of the recording, two people who are well-read and excellent listeners, and they both said it was still very high-level stuff. So rather than simply play this interview as it is and lose and frustrate a large fraction of our listening audience, let me take 5 or 10 minutes to give you the Coles Notes version of what was said. That way, many of you will have a framework in your mind, and you can decorate that framework with the specific details from our guests. And even if you don't find a place to hang some of their words, at least you'll have a partially decorated framework. And for those of you who find the conversation is just too much to keep up with, feel free to fast forward to the last 20 minutes of this episode, where our guests get out of the scientific weeds and talk at a more general level. And more importantly, they draw some really provocative comparisons between the evolution of proteins and the evolution of the Bible itself. In both cases, you have copies being made with modifications happening along the way and some kind of a filter that tries to hang on to the good stuff. Descent with modification and selection. It's the engine of evolution. Evolution of language, evolution of books, evolution of life. I guarantee it will make you think. So here's my summary of what our three guests said in this interview. First, the story of the flagellum is far more complicated and nuanced than what ID proponents typically present. The flagellum is not, absolutely not, something that can't tolerate any changes. It does not have to lose function when you alter the genetic sequence or leave out certain protein parts. In fact, there are thousands of types of bacterial flagella, all different in their genetic structure and in the proteins that make them up, and yet they still serve their purpose for their bacterial hosts. The second take-home message is the one that absolutely floored me when I first heard it a few weeks ago. It's a story with a recurring theme, and that theme is again the engine of evolution. a theme in which the gene for one particular protein nanomachine gets duplicated and the two copies then mutate differently. They diverge to produce two new different nanomachines with new functions that the original nanomachine didn't have. And then the genes for those new nanomachines might get duplicated and the two copies diverge to produce yet more, to produce yet another new nanomachine with yet another slightly different function. And then there's more duplications, more divergence, and new nanomachines coming into view. Again, the engine of evolution. So let's take that central idea of gene duplications and modifications and apply it in the story our guest told about the flagellum. They basically reverse engineered the flagellum, working their way backwards through those duplication and modification events, going backward through time, even to the point of the very earliest forms of life that ever existed. So they started with the flagellum, a nanomachine with a long whip that spins and propels the bacterium forward. There's a second nanomachine that looks very similar to it, similar genes and similar proteins, but instead of a long whip for propulsion, this one has a long needle that injects poisons into the prey of the bacterium. Both of those nanomachines have a third nanomachine built right into them, at the very core of their structure, a machine that pumps proteins out of the cell. Either the proteins that make the propeller to chase the prey, in the case of the flagellum, or the proteins that make up the needle that's used to kill the prey, in the case of what they call the injectosome. And in both cases, the protein pump works by spinning at high speed, like a spinning top, getting energy by burning an energy molecule called ATP, and forcing the proteins up through the central hole of that spinning top. That third machine, buried deep in the flagellum and in the needle used to catch prey, is quite similar between the two, but obviously with some important functional differences, entirely as one would expect if they both diverged from a common ancestral nanomachine. And those similarities between the two specialized protein pumps give us an idea of what that primitive ancestral pump might have looked like. A primitive protein pump spinning like a top and burning ATP. Those similarities and the image of the primitive ancestral protein pump share an uncanny resemblance to an entirely different nanomachine, a fourth nanomachine. One that has a history that goes back to the earliest forms of life. One that also looks like a spinning top, but this one doesn't burn ATP or pump proteins. Instead, it interacts with certain proteins to make ATP. This rotary engine, the fourth one, is running in reverse to make ATP. It's an energy factory, a generator. And then listen to this. Both the protein pump and the generator share an uncanny resemblance. They both look like a necklace made of 6 big pearls spinning at high speed around somebody's neck. They're both a 6-globed spinning top. The protein pump is a 6-glob top spinning forward to push proteins up through the center of the pearl necklace, while the generator is a 6-glob top spinning backwards that rotates a protein connected at the center of the pearl necklace, which then pumps out ATP molecules. And not only do the 6 glob proteins look similar between the two, but the arrangement of the various genes that produce those two different nanomachines are laid out in the same order. 2 completely different gene clusters, with bits and pieces of information scattered haphazardly across the chromosome, but both laid out in much the same way as each other, the same kind of haphazard scattering. It's as if the protein pump and the generator diverged from a previous ancestral gene, one that coded for a 6-globbed spinning top. And then we find there's yet another nanomachine, nanomachine number 6, one even more ancient than number 4 and 5. This one is a spinning top pushing an RNA molecule through the center of its necklace. Okay, well, usually people describe the spinning top racing along the RNA filament, but you say potato, I say potato. If you stop and think about it, whether it's the spinning top pushing itself up and down the filament, or the filament being pushed back and forth through the spinning top, we're really saying the exact same thing. And if your own jaw isn't already on the floor, then let me connect the dots for you. These incredibly different nanomachines all have the same fundamental underlying structure. A common structure that is so unusual, so unlike any other family of protein nanomachines. A 6-globbed spinning top, often spinning around a central filament, whether that filament is a strand of RNA in the case of the oldest ancestor, or a strand of protein in the youngest descendant, or a protein that turns a crank on an ATP energy factory in one of the intermediate forms. The resemblance is uncanny. It's like bumping into a stranger in the street, someone who has the exact same facial features that you have, and you start to ask questions and you find that you have the same medical histories, some of the same relatives, and you come to realize they were a family member that you never knew about until the data started to come in and questions started to be asked. So back to the origin story of the flagellum. This isn't yet a slam dunk case. Parts of the story are widely accepted by those who work in this corner of scientific research, but other parts are still quite speculative. But this emerging story has been generating scientific questions, and when those questions get pursued using the latest high-tech scientific methods, pieces of the puzzle start clicking into place. The body of data is definitely growing exponentially, and the image of the evolutionary journey is coming into focus. And if you ask me, this version of the origin story of the flagellum is more compelling and more exciting than the intelligent design version of that origin, and without all the theological, moral, and philosophical problems, especially the whole problem of malevolent design that Scott and I have talked about. So that's my version of the story we got from our three guests, scientific experts in the world of flagellum biology. If you're up to the challenge, listen to them for yourself and make up your own mind. But if you can't keep up, do not fear. Scott and I will debrief the whole thing again at a much more non-expert level. And if you're quick with sending us any questions or comments, we'll take those up in our conversation. So, for the more brave members of our audience, sit back and join me as we eavesdrop on the middle of my conversation with our three guests. And for the rest of you, fast forward to the last 20 minutes and join us there.
So that's one of the ways it's done. I don't do so much of that now, but yeah, one way is you stick the cell down and you stick the smallest possible bead you can down on a truncated propeller, or even just the hook below the propeller, so you can see it going round and round really fast.
What he means is, rather than trying to watch a nearly invisible hair-like flagellum spinning really fast behind the bacterium, you instead pin the bacterium down and attach a fairly heavy and large bead to that flagellum, and watch it spinning around much bigger, but a little bit more slowly.
Because then you can see the individual steps and try and work out sort of which molecule goes in at the exact moment it pushes it around, that kind of thing. That's how we learned a lot about it.
And I had to ask, putting a ball and chain on these poor, helpless little flagella, do you need ethics approval for that kind of thing?
Some people have asked me that before. So even before this, one of the early methods, so from the 1970s, was a so-called tethered cell, which is where you stick the filament down and the cell body goes around and around. And we do that still a lot. And the way you do that is you squeeze a solution of bacteria through a syringe. And someone did once ask me, is that cruel to the bacteria to cut the cells off? And, I was like, I kind of didn't expect it, but it got me thinking. I mean, there's philosophical questions in there, but certainly in Australia, I know for a fact now, there isn't ethics approval for anything up to insects. So like anything above insects would require ethics, but anything insects and below doesn't require it. And certainly E. coli is way below insects. so in that sense, I feel that we're completely covered there. But these things are actually quite fragile. It's worth understanding. The filaments break experimentally quite a lot. It's not like a submarine propeller. It's like potentially quite fragile.
So we've got the three of you here collectively representing decades of research on the bacterial flagellum, using almost every cutting-edge scientific tool known to science to understand that one family of proteins. So something tells me you're going to be able to shed some light and maybe some clarification on the bacterial flagellum. But maybe just to get us started, tell us why this family of proteins has merited so much attention, so much research. And why don't we start with Matt?
So why so much interest in the flagellum motor? I think it captures people's imagination. It's something simple enough that you can get interested in. It's like an outboard motor, right? Like a submarine. How does a submarine go? People kind of understand propellers. They understand rotary motors. I think there's general interest in that. And then if you look at it completely devoid of a context of life, it's impressive machinery. It operates fast, is the endpoint of chemotaxis, so helps bacteria swim to where their life is better. There's plenty of aspects by which you can be impressive in the nanotechnology sense. But it's a good question. I think there's plenty of other impressive things that get less notice. And so why the flagellum motor? I guess we are thankful. Maybe that's why we work on it. And we're certainly thankful for the attention, right? In that it gives us interested students and people generally are interested when we try and say, hey, this is important because of X, Y, and Z.
And Matt, those students, that research team that you've put together, you've recently made some interesting discoveries about the flagellum. Tell us about that.
So one of the fascinating things is that this amazing rotating nanomachine is itself powered by an even smaller rotating nanomachine. So that was only discovered a couple of years ago. We finally solved the structure of the ion channel that drives the rotation of the flagellum motor. And it turns out to be a 5 and 2, so a pentamer that rotates around a dimer stalk, much like some of the other things, the central stalk and something rotating around it. So now the race is on really globally to see this directly rotating in a cell or reconstitute it outside a cell and see it rotating. So I guess part of this is relevant because one of the things is, it's an amazing rotating nanomachine, but that's powered by another rotating nanomachine. And again, this type of rotating nanomachine is seen elsewhere. So not just in flagellum motors, it's seen in ion pumps and other exporting systems. So if we want to get down the speculation of the origin of the flagellum motor, whatever made the filament that's related to type 3 secretion or something that was an ancestor of those systems made what becomes the propeller, you still have to ask yourself, well, what drove the propeller?
Right.
What was the engine that drove the propeller around? And these kind of other rotating nanomachines, the stator unit, mod A, mod B, which is spread out in different systems, is a fascinating complex of its own right.
And Nick, why did you get into researching the bacterial flagellum?
Well, to be honest, Fred, I got in because it was back in the days of the fights about intelligent design creationism. And in the world of challenges to evolution, most of them aren't very good, in terms of many anti-evolutionists ask questions that have been answered, where all you got to do is look it up, or often many things 20 years ago, people had written rebuttals on them, but there wasn't, and 20 years ago, there wasn't much written, hardly anything written on the origin of the bacterial flagellum, even unlike some of the other systems that some of the intelligent design people talked about. It turns out on a subject like the evolution of the immune system, there actually, even 20 years ago, there were hundreds and hundreds of articles on the evolution of the immune system. But the evolution of the bacterial flagellum, there was almost nothing. And so that made it interesting, right? Somebody somewhere should look at the evolution of that system. And so I got into it, and honestly, as a bit of a hobby, and then got into it, eventually published with Mark Fallon an article on it that turned out to be one of the early articles on that subject. And so, and the work that is being done now, is an attempt to extend and test hypotheses in that space. And so yeah, I got into it for that reason, but it is the reason the creationists chose it was that it is amazing, right? It is a spectacular adaptation, and it's one that you can get your head around and the public can get their head around without having to know a lot of biochemistry, in a sense, because it's a thing, it sticks out of the cell and it spins and it makes the bacterium swim. It's made out of tiny little proteins. And so that's just incredible, right? So yeah, so that's how I got into it.
So last week we described the flagellum in detail, and we don't need to repeat that today, but I do want to confirm a few things with the three of you to make sure that what we learned represents the views of the scientific community, because some of the details can often conflict with statements that are regularly made by the intelligent design proponents. So, we learned that the flagellum is not just one thing, one entity with one precisely defined set of parts and a gene sequence that can't be altered in any way without losing its rotational function, which is their definition of irreducible complexity. Instead, there's a whole family of flagella, with dozens of members, hundreds of members maybe, all with distinct gene sequences and some
tens of thousands, hundreds of thousands, million. I mean, each bacteria that has a flagellum will have a slightly different sequence. There will be the occasional SNPs that differentiate one from another.
It's highly diverse in the flagellum motor, so they're all chopped up all over the place. So they're all spread out, which is proof of how much chop and change and horizontal gene transfer there is,
right?
Because they are not the same in most places. Yeah.
So there's a whole evolutionary history there.
I mean, it's a bit like when people talk about the human genome. There's no such thing as the human genome. There are, 7.5 billion human genomes, because they're all different. And it's the same with flagella. Every strain will have a slightly different set of genes. And in those genes, there will be mutations. Some of those mutations will make differences to the amino acids in the proteins. Almost all of those differences in the amino acids won't make any difference to the function. but nonetheless, they are all different from each other.
Right.
There's not one thing that's come down from on high. That's the bacterial flagellum.
And more importantly, some of them can be missing parts and still maintain that function.
Exactly.
Which is what we hear from ID, that can't happen. So they still work.
Many are structurally very different as well. So at the sequence level, you have difference, but also where those sequences are located in the genome are very different in different genomes. Some are all close together, some are far apart. And structurally, if you took a zoomed out view, you would see whole blobs that were there or not there in different motors. So it's a good point to mention how diverse they are. And so obviously some of those ones with more blobs, there are other motors that don't have those blobs. So it would show that those blobs are not essential, right? Because we know here's another motor with a few blobs in certain places and it still rotates perfectly fine.
Right. Good. Now, we also learned that the flagellum is not just a rotary engine with a big whip, the propeller, but that at the very deepest part of it, at its base, there's another machine, the type 3 secretion system, a machine that squirts out proteins, in fact, the very proteins that create the flagellum, and that there's another bacterial machine, a third one, the injectosome, which also has this type 3 secretion system at its base. Only this one squirts out a different set of proteins, the ones that create the needle and then the toxins that they use for the bacterium. So 2 very different machines mounted on top of a third machine. So Mark, you studied the system directly using a blend of genetics and I think some biophysical techniques as well. Is this a good summary statement?
Yeah, exactly. I mean, this is a modular setup so that you can have the type 3 secretion systems that have many essential conserved functions accompanied by additional things, the flagellar filament or the injectosome. And it is this modular design that's at the heart of this, and you can swap these components around in some ways, so you can take one flagella system and put a different flagellin in it, and it will still work a lot of the time. It's been shown with the injectosome that you can take one of the antigenic variants that's usually secreted and replace it with another one, and it still gets secreted. So this whole thing, I mean, particularly with the type 3 secretion, one of the things I showed was that it was just this mind-blowing diversity in that what you have is effectively a molecular syringe that will squirt these proteins out typically into eukaryotic cells. But the repertoire of proteins that go through the system will vary from one strain of E. coli to another quite dramatically. And they are not all in the same genetic locus as the genes for the structural apparatus. They can be scattered throughout the chromosome. And they're often carried on mobile genetic elements called bacteriophages, bacterial viruses that integrate into the chromosome.
Remember, this is exactly what Dr. James Shapiro told us 2 weeks ago when he presented a whole new set of genetic mechanisms that explain evolution. Cells moving whole chunks of DNA around.
So the idea that there is some kind of, type of, you could say this is the thing that is the typical of the flagellum of type 3 secretion or whatever, is misleading. And it's one of the, Stephen Jay Gould used to make much of this, that what we see in biology is just riotous diversity. And for a physicist, they can say that every proton in the universe is more or less the same because they're all kind of the same thing. Whereas, any entity that we see in biology is just one example of riotous diversity that you see for that particular entity. Just as humans are all different from each other, each of these systems is different. And sometimes the differences are very large and they're big and functional. Sometimes they're very small. But this is
right.
Okay.
Now I want to bring another fourth player into the story, another bacterial machine, the ATP synthase, which would have to be, I'm guessing, one of the oldest bacterial machines because it creates ATP. And what doesn't a cell do that doesn't require ATP? But more to the point, this fourth machine has some amazing jaw-dropping similarities with the type 3 secretion system, or so I gather from the paper that Nick and Mark, you wrote. Similarities that give us reason to think that they descend from a common ancestral gene.
Oh, most definitely. I don't think there's any questions about that now.
And Nick and Matthew?
I would just briefly say that is an example of an evolutionary hypothesis that started as there was sort of a little thread of evidence, right? And the first piece of evidence was maybe discovered way back in the 90s or late 80s, which was that a gene in the ATPase and a gene in the flagellum, the proteins have some detectable similarities in the sequence, right? And so that was known for a long time. But then it wasn't until the 2000s when it became apparent that not only was the sequence the same, that these two proteins kind of had basically a very similar structure, and they probably both formed a system of 6 proteins in a ring. And so that 6 protein structure was shared between the flagellum and this ATP synthase. And then it was actually Mark, I think, that
second stalk,
added another piece of evidence, which is that the external stalk that attaches to that 6 protein system is also shared between the two systems. And then there were a few more papers around 2010, 2012 or so, I think, that showed there's an internal stalk that's also shared between the two systems. And then just recently, a paper that myself and Matt Baker and some others were on showed that those 3 proteins, typically the genes for them are in the same order on the chromosome across these different systems, across the flagellum and the ATP synthase. And that suggests that whole, what we would call a synteny, the 3 genes in the same order, seems to go all the way back to not even the last common ancestor of life, well before that, because there was some ancestral system that had those 3 genes, or maybe there were 4 at the time. Some of them may have merged. Those several genes in the same order on a chromosome back well before the last common ancestor. And there's some other evidence too that suggests that these things are what we call homologous, evolutionarily related. And so that's a piece of evidence. That's it's sort of an unnecessary similarity, right? And these unnecessary similarities are little bits of evidence that we call homologies that indicate shared ancestry. The reason they're similar is because they were copied from an ancestor. And this was just a particularly spectacular example that goes all the way back. That's a case where the evidence accumulates in a specific hypothesis for a part of the flagellum. This isn't the whole flagellum, just a core part of the secretion system, actually. It goes way, way, way back, and clearly, didn't have the flagellum function originally. It was doing, and there's a whole debate about what this says about the origin of the ATP synthase and how did that all come together. But parts of this system trace back to very ancient origins that weren't originally involved in motility. Those other 20 flagellum proteins have different origins, and we could talk about those if you want. And so, I remember debating with the creationists, and they were sort of scoffing at some of the speculations. And in this particular case, the evidence is really accumulated to kind of firm this hypothesis up.
Right. And I understand that these clusters of genes, the one cluster for the type 3 secretion system and the other cluster for the ATP synthase, they're on completely different chromosomes. When Nick gives his answer here, he'll refer to the 3 main forms of life on Earth: eukaryotes, bacteria, and archaea. The current understanding is that the very, very earliest forms of life were simple single-celled forms which could be divided into 2 groups, either archaea or bacteria. Billions of years later, an archaea bug combined with a bacterial bug, or the other way around, and put together their evolved complexity into a much more complicated form of life called eukaryotes. And those eukaryotes were the ancestors that produced all the plants, animals, and humans that we see today.
You may be thinking of the following, which is that a eukaryotic cell has an ATPase in its mitochondrion, which is derived from the bacterial ancestor of mitochondria. And then it has a second ATPase in its nuclear genome, which is closely or more closely related to the formerly archaea bacteria, another big group. And that's in the nuclear genome. And those 2 systems are related to each other. They're closer relatives. And then the next closest relative to all of them is the ATPase in the flagellum type 3 secretion system. And so you can, once you start to put this together, you can give an interesting talk where you tell people, like, okay, there's this protein that's in your mitochondria providing you with energy, but there's another one doing a pumping function. It's in your nuclear genome. And there's another one in the bacteria, and they're using it to help build the system that they're swimming around with, which is the flagellum. And then some of the other ones are using that protein in a secretion system to attack you. And all of this is going on at the same time, and it's all using these in part related systems, right? And so, you can tell some interesting stories that way. So it is
one thing that Darwin made the analogy that with language, that the way biological systems evolve is similar to the way languages evolve. And in languages, we often get multiple words that have the same ancestor that have slightly different meanings. And it's like, so we have in English, for example, we have the word lawful, we have the word loyal, we have the word legal. They all kind of have common ancestry, but they, over time, they diverge. So the word legal and the word loyal actually have slightly different connotations. They have different functions in the language. And it's the same kind of process in biology that you get. If you get a duplication, so you end up with two copies of a thing, they can diverge in their functions. But even if there's just one copy in the genome, over time, its function can change in one lineage compared to another lineage.
Okay.
This is a very common thing that we see. And it's one of the things that explains how the flagellum evolved, is that you had a series of gene duplications where the actual components that form the kind of filament, there was a common ancestor that was just one thing that, was a generalist and did this general job. And then with gene duplications, you got more specialized parts that sit in different arrangements within the spatial orientation.
Right.
And that's the thing that we're seeing it when we see these divergences in sequences, and we then detect sequence similarity, what we call sequence homology. It's all effectively one process. And if you're going to say, well, when we look at the flagellum or flagella in general, flagella systems, we can explain this because something special happened at a certain stage. You sort of think, well, hang on, you have to accept that if we've got two different E. coli strains and they show sequence similarity, but they're not identical, you've agreed that they've diverged from a common ancestor and they've acquired mutations over time to become like this. And then if we look at E. coli and Salmonella, well, the same processes are going on. And then if you go far enough back, as Nick is going, and say, well, if we compare archaea, ATPases and archaea with those in bacteria and those in eukaryotes, we see similar divergence. It's all one kind of seamless process. Where do you say, oh, we need a miracle to explain this? It's back to this old idea, natura non facit saltum, which is that, nature doesn't make leaps. There's no place where you say, wow, this is just, there must have been intelligent design at this moment because that difference between that and that is just a miracle. Right. We don't see that in these systems.
Right. So I want to look more closely at that evolutionary history. I just want to go back a few more steps, a couple, maybe a billion years, and then put that all together, that whole sequence of events that tells us how flagellum came to be. So the type 3 secretion system, we talk about the similarities between it and the ATP synthase, and there was a suggestion that those came from an ancestral gene, a common ancestral protein product. I understand that from your paper that the one suggestion, maybe there's evidence to suggest that ancient ancestor was an RNA-related, an ATP-dependent RNA transporter, helicase.
Oh, so that's where you're really getting into it there. So for that specific protein, there's the ATPase, or ATP synthase system, and then there's this big protein that we call an ATPase that's, the sort of the biggest protein in that system. And it has additional relatives beyond those we've discussed. And one is REC-A, which I think is, an RNA helicase, I forget. But those proteins also form a ring of 6 copies, and then they process something through the middle.
So just to emphasize the similarity between the two kinds of proteins and to put an image in the mind of the listener to help understand what Nick is saying here, and which will come up again a few more times in this episode, both the RNA helicase and the type 3 secretory system are made up of 6 globs of proteins arranged in a ring structure, wrapped around a central filament that they're working on. A filament of RNA in the case of the helicase and a filament of protein in the case of the type 3 secretory system. And this 6-globed ring isn't just wrapped around the filament, but it's spinning around that filament as it moves from one end of the filament to the other, which is the same as saying it spins in a circle as it moves the RNA or the protein through the ring. Whether the motor is moving relative to the filament or the filament relative to the motor, it's the same thing. The helicase spins around the RNA as it moves itself along the strand of RNA, while the type 3 secretory system spins in place as it moves the protein forward. It's a very unusual arrangement, and so the similarities between these two really make one wonder about a common origin.
And so probably that was an even more ancestral function. And then in one case, this became membrane bound, and that was maybe the common ancestor of the ATP synthase and the sort of core part of the type 3 secretion system. And that common ancestor, it was probably processing something with that ATPase, but processing it into the membrane. And it's actually not clear. It could have been kind of a secretion system at that point. And then perhaps one version of it got stuck and focused on, instead of burning ATP to pump something, it wiggled something to create ATP, for all we know. It could have been a bit of a reversal of a process. So there are some papers exploring that exact issue. But yeah, that ATPase protein goes way, way back, and there's even more. It's one of the most conserved proteins, I think, around. And so there's more and more distant relatives doing various things. But you can kind of see at least a few steps of adding complexity in terms of making this protein part of a larger system and then part of a larger system again. And it just happens to be this is one that is conserved enough you can trace it back in a more distant way. But yeah, these are all groups, so it's hard to say that, what the ancestral function is, but you can say that there's several sisters that are doing somewhat similar things.
So if I can boil down, Nick, what you just said for the listeners again, because these are non-experts, you said that we're talking about three different enzymes. We were talking about the type 3 secretion system and the ATP synthase and how they may have derived from another enzyme, this RNA transporting. You talked about REC-A and other REC enzymes. All three of them have this common structure. You talked about 6 copies. You mean 6 proteins, 6 globs arranged in a circle, and they interact with some molecule up through the middle of the circle. In the case of the RNA transporter, it's interacting with RNA, and maybe there was a slight, a mutation, a modification to that enzyme. Now it recognized more proteins, which is where you get the type 3 secretion system. And the interaction with the proteins also brings in the ATP synthase, because that ATP metabolizing enzyme is now interacting with the synthase portion of that enzyme. So that's what I heard.
So something like that could have happened. I wouldn't overstate our knowledge, right? There is a paper that explores this issue in some depth, and I think their hypothesis is, all you could say is that maybe this hexameric protein was involved with processing chains of things through it. That might be what it is.
The attraction of helicases is that it's a link. Like rotary motors are pretty rare. So the flagellum motor is one, ATPases are others. The ones you're talking about now, the archaeal motor is similar to the flagellum motor, but i.e. it rotates, but otherwise very dissimilar. And a helicase is maybe somewhere that gets you towards something that rotates because it's moving on a helical track, and thus it kind of rotates. Plus, it's got this 6-mer symmetry that you highlighted. But I think I'm certainly not aware of like real smoking gun evidence that say helicases predate the type 3 secretion system and are the origin of the type 3 secretion system. I'd be happy to hear it more about that, but I think that is quite close to sort of nice hypotheses that line up in a lot of ways, but I'm not sure we have the evidence for that.
Right. Okay, and then to finish that evolutionary history, then the ancestor to the type 3 secretion system that came out of this that we're just talking about, then further mutated so that one became more selective. It responded more to the bacterial flagellar proteins, and it started to squirt out those, and you have now this type 3 secretion system at the base of the flagellum, whereas the ancestor could have mutated in a different direction, been a bit more specific for the needle, that hypodermic needle that you talked about, and the toxins, and it starts squirting those out. So that I guess I
wanted to hear about what Mark had to say on this, because for me, I wasn't sure we knew for sure which one came first in the broad sense, whether it was a type 3. We don't know, I think, for me, whether it's type 3 secretion system becoming a flagellum or the part of the flagellum that's type 3 secretion becoming the injector zone.
And then there's the argument, it's like, humans and monkeys have did humans come from monkeys? No, we came from a common ancestor that you might say was a monkey if you saw it on the street, but it isn't the same as monkeys. It's the same argument. Whatever it was the ancestor. It had some kind of, we can wave our hand and say it had these functions. But it's not the same as anything now. It's not going to be a type 3 secretion system or a flagella system. It's going to be a precursor.
I thought the type 3 secretion system was at the heart of the flagellum, and it squirted out the proteins of the flagellum proteins.
Well, yeah, so the commonalities you find in type 3 secretion systems associated with flagella and the commonalities you find in the other type 3 secretion systems that are found that are moving proteins into eukaryotic cells, you can see they're in a different functional context, and they are separate lineages. But to go back and say, what did the ancestor do? Did it actually put proteins into eukaryotic cells? Did it make a flagella filament? Or did it just do something else, very simple, like exported proteins from the cell? I don't think we can say. I think one of the issues is that in science, we have degrees of certainty. And we talk about everything. We test a hypothesis, we propose a hypothesis, we test it, we try and refute it. And sometimes we get to the stage where actually it's a fact. We all accept that atoms exist. There was a time when the existence of atoms was a hypothesis. Now we say that atoms exist, that's it. This stuff is still, the idea that these have a common ancestor, I think is a fact. It's getting close to a fact now, pretty much. But what that ancestor did, quite how it worked, that's still in an area of uncertainty.
So if we can't say that the flagellum came from the type 3 secretion system, can we at least also say that the type 3 secretion system didn't come from the flagellum? ID proponents will often say that the type 3 secretion system is a devolved flagellum.
So I've read all of that, and I think it's fair to say that the most recent and best paper, which is now honestly probably 10 years old, but that exact question has been subjected to a phylogenetic analysis. And the result that paper got, it was Abbey and Gofna, I think. The result was that the quote-unquote injectosome, we should be more specific, the type 3 secretion system, which is specialized on attacking eukaryotes, appears to be phylogenetically nested within flagella to the limits of the techniques and the sequences that they were using at the time. I would say, having read that paper closely, I would say that might not be the end of that discussion, but that is the result that they got, right? I think, I forget the exact numbers, but it was something like 75% probability maybe. It would be very interesting to have another crack at that one and see if the newer models and newer data sets, if you still get that same answer. But that is what that result was. And so I think that's fair to say. I don't think there's an issue, though. Like when you say the type 3 secretion system, what do you mean? Do you mean that specific injectosome, right? Or do you mean essentially the combination of the 10 or so core proteins that combine to create that function of secreting proteins in that manner? The reason it's called type 3, right, is there's other secretion systems out there. There's type 1, type 2, type 4, type 5. I think you're up at 6 or 7 now.
Oh, hi.
Right? So there's a whole number of secretion systems out there. Several of those secretion systems are known to, in some cases, stuff that gets secreted gets stuck on the outside of the cell, making something called a pilus, right, which is just a filament sticking outside the cell. So several different secretion systems have added on that capability. Type 3 secretion systems are one of those. And in one other instance, a type 4 secretion system, there are versions of that produce pili, these things that stick out. There are versions of that system that do motility. There's something called twitching motility, where this pilus is stuck out and retracted, and this is used to move a cell around. And then there's a version of that actually turns the pilus around and creates another flagellum that's called the archaeal or the archaebacterial flagellum. And so there's another entire instance, which the intelligent design people don't mention very much, of a different secretion system related to a different motility system that produces a filament that sticks outside the cell and spins, but it's completely evolutionarily not related to the bacterial flagellum that we're talking about. And in that case, it so happens that the phylogenetics that I've seen puts that archaeal flagellum nested inside of a couple of these secretion systems quite nicely. So in one case, you've got the available phylogeny telling very directly the story you might want to see if you wanted to have kind of everything be in a nice phylogenetic order. In the case of the bacterial flagellum, you don't quite see that. and the intelligent design people get very focused on that bacterial flagellum one, but I don't see that as that significant of a point. The big story, right, is that secretion systems build things on the outside sometimes, and sometimes additional things get linked onto those systems and produce motility. I think the existence of this non-flagellar type 3 secretion system is a piece of evidence for that general story, even if that specific phylogenetic relationship currently makes it appear derived. Maybe that story will change a little bit. Probably not. Probably that's where it is. And it's not crazy, right, to think that the flagellum in some cases take on a reduced function, because we know of other cases where bacterial flagella have lost some of their parts and have sort of re-specialized on some sort of secretion function, where they only retain the function of secretion. So I think it's Buchnera aphidicola, which is a bacterium in an aphid. It's covered in sort of the basal bodies of flagella, but it doesn't appear to be motile, and it's probably using those basal bodies to secrete proteins that interact with aphid cells in some way. And so it's not crazy at all to think that you'd have a reduced function, and maybe the classic non-flagellar type 3 secretion system is just a particularly successful example of that. But I mean, the one other important point that the existence of that system shows, right, is that you don't need the entire all the flagellum proteins all together at once to produce a selectable function. You just need a subset, and there's various functional subsets that seem to have other functions, right? And so that point is established as well by that. So yeah, getting hung up on that phylogenetic question, I've never sort of understood exactly why the ID people latch onto that, except that, they got to latch onto something to kind of keep the gig going.
Well, presumably they're saying that there was a miraculous event that made the flagellum, and there was this one event, and that therefore can be used to explain type 3 secretion if it's a break. But I mean, the problem in evolutionary biology generally is that the relationship between simplicity and complexity is not itself simple, so that sometimes you can have evolution in the direction of a much more of a complex system, but then in a particular environment, that system then loses some of its components and becomes simple again. And, we see that with parasites quite commonly, that they lose a lot of things that they don't need anymore in a particular environment. Even flagellomotility. We see in E. coli, we have flagellomotility in many strains, but in a subgroup of E. coli that we call Shigella, they have lost their flagellomotility. We don't still know exactly why, but there's clearly some selective pressure to get rid of it. But we can see they had it because we see the smoking gun of some flagella genes still left in their genome. They're just not functional anymore. And so these relationships between, the direction of travel, what I would say is that you can look at, if you take a complex system and then you look at a simple derivative of it, you can say, well, there is a simple way of doing this, and that could be the way the ancestor was. It gives us a feeling of how a simpler ancestor might be, even if it isn't, in this case, an ancestor. It's actually a derived form that's simplified. But it shows that those simpler progenitors that we would hypothesize to be along the route to complexity can exist and can function and can do things. And in conceptualizing all that, I think that's an important step, an important facet of the argument there, that it's not an inevitable move towards complexity. Sometimes you go towards complexity to come back to simple, and so forth.
Right. One thing I'm hearing from both of you is that there is room for interpretation of the data. We have we're gathering a greater and greater body of data, but there's still not enough to really be definitive, and so there's a lot of room for interpretation. And different scientists will interpret things in different ways, but then you've also got how the intelligent design community have interpreted a lot of the data. Is it fair to say that there's that room for interpretation here?
Oh, it all depends what you mean, right? There are quite narrow questions, right, to get debated in terms of, one could be this question of what is the exact phylogenetic positioning of a couple of different systems, right? And those are pretty narrow technical questions. Sometimes all the data points in one way and boom, you're basically done and everyone's convinced and it's kind of over. Sometimes it's ambiguous. Anything that happened billions and billions of years ago and where the data that you've got is down to a handful of proteins and whatever sequences have been preserved, right? That's a tougher situation than, many systems.
Right.
If you look at the evolution of the immune system, instead of being perhaps 3 billion years old, that's maybe only 500 million years old. And we have a lot more intermediate steps preserved in the phylogenetic tree than we might. I mean, the bacterial flagellum, for all we know, it could have been in the last common ancestor of bacteria, right? There's at least one paper that does a study that suggests that, right? And so it is as old, perhaps, as bacteria in general. And there are a great many unresolved questions about what was that common ancestor of bacteria like. And it's quite complex for a large number of reasons. One of the hardest questions in evolutionary biology, right? Stuff going back to the last common ancestor of bacteria. So in that sense, there's a lot of unresolved questions, and I could name a couple other specific debates if you're interested. But I don't think that range of debate extends to maybe there were miracles involved somewhere, right? That's a very ambitious hypothesis.
Yeah, I mean, this is the nub of it, isn't it?
Yeah.
That there is levels of certainty and uncertainty in science, and that changes over time. Sometimes things that we thought we knew, we turn out that's not true, but other times this is no longer a theory, this is fact. Atoms exist, cells exist. Nobody questions, DNA is a hereditary molecule. Before Crick and Watson, people could have argued about that. Now we have a much more sophisticated understanding, and this is not just science. This is scholarship. And if you want to turn this argument back to these people, you say, well, look at the evolution of Gospels. Look at the Gospels that you read in your Bible in English didn't fall out the sky. They have ancestors. They have ancestors in English translations. They have ancestors in the original Greek. But nobody's got the original manuscript that Luke wrote or John wrote or Matthew wrote. We have various bits and pieces, and you can work out what the common ancestor of all those bits and pieces look like to try and infer what that manuscript was that Matthew or Luke or John wrote. You can look at the relationships between those Gospels, and you can infer similarities that we might fancifully call homologies between sections. So we can say, there were bits of Mark that we will see in Matthew and in Luke that look as if they have a common ancestor. There are more stretch hypotheses. There are bits in the Gospel of Matthew and the Gospel of Luke that look as if they have a common ancestor that's not in Mark, and there's this Q hypothesis that suggests there was a common ancestor there. So this evolutionary way of thinking is not just restricted. I've mentioned already languages, but it also applies to manuscripts. It even applies to the manuscripts of the Bible. That we don't, and this levels of inference and certainty. There's a spectrum there as well. So we could argue that, but you could even argue. I mean, was there a one draft of the Gospel of Matthew, or did he make multiple drafts, multiple copies, and they all varied slightly? All came from his hand, or did he have one draft? We don't know. So this kind of thinking and this kind of uncertainty is just part of scholarship. You can't. I did give a talk at a New Testament scholars convention where I made these analogies, and I said, we can even make analogies. We talk about the protein world now, but we then talk about the RNA world. Your Gospels were written in Hellenistic Greek, but there's a substratum of Aramaic because Jesus spoke Aramaic and all these actors spoke Aramaic in their day, and you can see the Aramaic substratum. So these ways of thinking we're not being fanciful as scientists or bizarre or whatever. The same ways of thinking apply across multiple domains of scholarship. Same arguments about languages. Are all languages coming from a common ancestor? We can see that all the languages that we call Indo-European have a common ancestor. We can say, oh, there it is. Oh, there's Hittite coming. That places the common ancestor further back. But then can we say, well, actually, there is a common ancestor with other language groups? Do they all have a common ancestor? Then we have to throw our hands up and say, well, that's very speculative. It's hard. I mean, in life, we know that all cellular life has a common ancestor. For languages, we don't know whether there was ever a proto-world.
Mark, I just have to say, I'm absolutely amazed you were a speaker at a New Testament scholars conference.
Well, I used to work with a guy who did all this stuff, this phylogenetic inferences on gospel manuscripts.
Okay.
There's a whole movement in the humanities where digital humanities, they call it, where they look at the evolution of manuscripts, not just New Testament manuscripts, but what version of Ovid or versions of Chaucer, what was the original version of the Canterbury Tales? Was there an original version, or did you do several drafts? This is all it's all out there, and it's interesting. It's a common thing.
there's, I'll use the magic phrase, right? It's the commonality between evolution and these other fields, which is descent with modification.
Exactly. And that's the number. And Darwin pointed that out, these analogies, even in the origin. Yeah.
Anytime you have a history of copying, occasional changes, then you get this descent with modification pattern. And then you can, either informally or even statistically try and estimate that copying history. And you can start to put statistics on it and levels of confidence, given different things. So it's I did a paper. It was called "The Evolution of Anti-Evolutionism," right? And I copying history of anti-evolution bills because they'd been copied. Now, I mean, you could, if you wanted to be the clever, intelligent design advocate, right? You could say, "Oh, well, descent with modification is happening there, but there's still an intelligent designer involved in those copying histories, right? In the copying histories of documents and things like that." And that's, if you wanted to be clever, you would go that direction. And I would say then you're back to the plausibility arguments about, well, what do we know about the processes that change these different kinds of sequences, right? We do know something about the processes that change documents, and so it's reasonable to conclude that humans are involved there. Some changes, it's interesting even in that situation, right? Some of the changes that happen in documents are, a deliberate editor deliberately doing something. Other changes are accidental, and sometimes you can tell the difference between those. Copying mistakes, typos, etc.
When you're talking about handwritten manuscripts, accidental changes are very common. They skip a line or they...
Even in right-wing legislation in a digital form, many of those bills get sent around and they get stuck in by some staffer and they make a couple of edits because, they want to avoid some potential future lawsuit or something, and you end up with something that doesn't actually make sense if you were to write it, from the beginning. It wouldn't really make very much sense. But so you get, and that gets copied just because it's sort of a bit of an automated activity. So whereas in evolution, that's the exact argument about natural selection and mutation and all the things we know about how different kinds of changes.
Well, these arguments can also, 13, 14 years ago, a colleague of mine, Barbara Baudelaire, we played these arguments out about the origin of species. So we talk about Darwin's Origin of Species. There was no such thing. There were 6 different editions, and about a third of the text changed between the editions. And she actually made, effectively a phylogeny of the text and showed that how the text changed from one edition to another. Some of those changes were deliberate, obviously. Some of them were probably just typos. Why would he change a comma or something like that? There's no reason for it, but you can see. So this is the point, that this way of argument is common across a lot of scholarship. It's not something, and uncertainties about these things. Uncertainties in part of life, and
I never would have expected this episode on flagellum evolution to turn into one of the evolution of the New Testament scriptures. I didn't expect that.
You have to turn the argument and ask them.
Yes.
This process of evolution has gone in the present. Descent with modification, where some of those changes may be functional, where it changes the meaning. Many of them are not, but they nonetheless have a phylogenetic signal. So if you've got a bunch of things that have a particular change, you can say they belong together on that lineage. But it gets more complicated because you have the equivalent of horizontal gene transfer, where they can copy a section of one manuscript into another manuscript and obliterate the phylogeny. So it's fascinating that these problems are at their heart the same problems in different lines of scholarship. And
we did speak to an ID advocate a couple of weeks ago, and Scott and I pointed out that the science proceeds by making and testing hypotheses, making predictions, collecting data, doing the experiments, the kinds that you're talking about. And we said that we don't really see ID people doing any of these kinds of things to test the questions that they're proposing. And the response that we got at that time, it caught me off guard. His answer was that secular scientists are doing all of that, these things that we were saying the ID people need to do. In fact, his exact words were,
and I think that's what a lot of laboratories are doing. That's what they are doing. They essentially work from a design perspective, but they don't call it that. They go on and say, well...
Is that a fair claim? Do you and your colleagues find it hard to do experiments and even talk about the findings without alluding to purpose? This was made for that purpose. This does that thing. Allusions to design. Richard Dawkins talks about the appearance of design. What are your responses to that?
So the assertion is that we whether we know it or not, we sort of think there is a designer, and we're looking to elements of that design. So I don't think that's the case, but it's kind of like sloppy language, and I try and hope my students get the importance of precise language. So it's very easy to think of purpose just from a bacterial point of view. Very commonly, I say, and others would say, well, the bacteria get hungry and then they look for food. So they go swimming over there, right? That's kind of true, in that it's like local nutrient is depleted. It's swimming along a concentration gradient in time towards or away from food. There's a sensory system that can interpret that and change the direction of the motor. Those sort of mechanical and chemical features are true, but there is no intent, there is no purpose. And with sloppy language, we start to sort of get familiar and personify the bacteria. So I think similarly with the purpose of a protein, it's just it's seeing correlation and assuming purpose where there isn't. But Mark,
this goes back to Darwin's time, and Gillian Beer has written extensively on this. Darwin himself, when he chose the term natural selection, he got told off. Like, hang on, you're implying there's a selector. There's agency doing something, and he in one of his letters he said, perhaps I should have called it natural preservation or something like that. And in the closing words of The Origin, he said originally breathed. He used this term, invoking the idea that there was some kind of. And Gillian Beer spent a lot, trying to say that it's very difficult. It was difficult for Darwin when he was writing it. It's still difficult today to pull back from the idea of agency in nature, and we see the appearance of design, but it isn't designed in the way that an intelligence designs it. It comes about through the process of natural selection, descent with modification, and it's a difficult one.
So ultimately, I'm a synthetic biologist, and the paradigm in that field is design, build, test, right? And that really is like me as a designer saying, I want to take that protein from that system and that protein from that system, try and cobble them together to get them to do something new. That definitely has design intent. But what we try and do is learn about the complexity of life and how these difficult systems work with each other, which took billions of years and has a very complicated history of co-interaction, which is why it's so hard for me to take two proteins and design them to work together. Right. Phenomenally difficult, right? And so in that aspect, yes, I try and design things, and I learn it's really, really bloody hard, but life all around us has done this with the marvelous computer of evolution and natural selection.
So can I build on that, what you just said there, Matt, and also, Mark, what you were saying? Could we say that the cell itself and evolutionary pressures, they are the designer with a lowercase d, that cells are using old parts and recontextualizing them, they're building machines on top of machines, they're arriving at a genetic or a cellular solution to an evolutionary pressure, and solution is just a different word for design. And then they mutate through that as new pressures arise. We did an interview, we played it again last week, where we listened to Dr. James Shapiro, a geneticist at UChicago, and he told us these kinds of things, that the cell he gave us a whole new perspective on the way genes work, how they're shuffling parts of genes all over the place and putting things together in response to evolutionary pressures. So can the cell itself be an inanimate, impersonal designer?
It's not the cell, it's the process of natural selection is what is. I mean, the cell, you can argue that the cell is trying to just survive, and it will have a number of systems that adapt in real time to the environment around it to ensure that it continues to survive. But this business of why do we see the appearance of design in nature is due to the process of mutation and then selection. And it's natural selection is the driver then. So
and at the population level, when we think of the cell as the designer, we're talking about one cell, and one cell can't go, hey, this protein's a bit better than that one, right? But if we have billions and billions of cells with each slightly different ones, then at the population level, the optimal answer, can survive.
Right. Well, we really should leave it at that. We're not going to, who knows where it's going to go from here. I do want to thank all three of you for coming and giving us your expertise to bear on this debate about the flagellum. And you brought a lot of clarity to something that I found a bit confusing hearing solely from the ID perspective. So I want to thank you.
There was one, can I just make one more point?
Sure.
which is the idea that the flagellum is this wonderfully elegant device. That's one perspective. If you're a young woman suffering from ascending UTI, where an E. coli is crawling up from your bladder into your kidneys and into your bloodstream and making you ill, and potentially, if you're old enough and frail enough, it might kill you. The flagellum's doing that. It's helping to do that. So this is not a neutral kind of elegant thing. It has a role sometimes in maintaining a healthy microbiome, perhaps, but other times it's an agent of disease. And
right.
So is the intelligent designer, if such a thing exists, benign? Can we divide? Can we derive that hypothesis? That it's, it's just a bit of it's an odd situation I have.
And we did discuss that. My co-host and I did discuss that with the ID advocates that we brought on, this idea of malevolent design. We did talk about that. Yeah. So a lot of fun. Again, thank you for coming on to the episode. I really appreciate it.
Thank you, guys. Thanks.
Thanks. You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.